So the topic of the lecture is causative agents of viral hepatitis. Viral hepatitis are a systemic diseases primarily involving the liver. Most cases of acute viral hepatitis in children and in adults are caused by one of the following agents: hepatitis A virus, HAV, hepatitis B virus, HBV, hepatitis C virus, HCV, hepatitis D virus, HDV, hepatitis G virus, HDV, and hepatitis E virus. It was someone hepatitis F virus. Did you notice the F is missed? It was isolated once and it was not studied well, but the next virus that was isolated was named G. So maybe in future we will know about, we will study hepatitis F virus. Hepatitis viruses are transmitted, so by the name of disease it's clear that these viruses are able to reproduce in hepatocytes, the cells of liver. Reproduction of them is going on in hepatocytes. But these hepatitis differ by epidemiology and by the transmission ways and the pathogenesis of infection. They differ by epidemiology. So hepatitis viruses are transmitted by two different routes, either by blood, parenteral transmission, any contact with blood, and these viruses are circulated in blood, and parenteral means avoid the digestive tract. They don't go through digestive tract. They don't reproduce in digestive tract. And other viruses are transmitted by fecal oral route, exactly enteral transmission. Viruses those go through digestive tract. So different mechanism of transmission, routes of transmission. Blood-borne viruses, parenteral mechanism of transmission. What are they? They are hepatitis B virus, hepatitis D virus, hepatitis C virus, and hepatitis G virus. Exactly, they are connected. Then I will explain why B and D and C and G, there is connection between them. Viral hepatitis with parenteral mechanism of transmission, hepatitis B, hepatitis C, and hepatitis D. Hepatitis G is not studied well anyway. It is named. Potentially any situation, including contact to infected blood, can result in infection. So blood contact mechanism. For the infectious processes caused by such viruses, typically is the chronic current of diseases and carriage of viruses. So some patients has clinical symptoms if they are infected by these viruses. So they are, in this case, we are talking about chronic clinical form. And some people are just carriers of the viruses. Carriers are infected by pathogenic microbes, but they have no clinical symptoms of disease. So the difference between chronic infection and carrier state is that carriers have no clinical symptoms. Sometimes they are more dangerous than patients because of carriers they don't know that they're infected. They have no clinical symptoms. They have no complaints. They are not isolated. And they communicate with other people, and carriers are able to infect many people. And such infectious disease caused by these viruses, they are very dangerous because of finally reproduction of these viruses leads to destruction of hepatocytes, liver cells, and cirrhosis is developed. And these viruses, hepatitis B and hepatitis C virus, they have oncogenic properties. They are able to cause malignant transformation of the liver cells. So the primary liver cancer is developed often in patients with this hepatitis compared with other people. Those are not infected by these viruses. So they have oncogenic properties. Viruses with a fecal oral transmission mechanism, they are hepatitis A virus and hepatitis E virus. They are transmitted by fecal oral route. Viral hepatitis with fecal oral route of transmission, hepatitis A and hepatitis E. You know ways, it is just route. What does it mean? Means these microbes go through digestive tract and they go out by feces, and they are able to survive in environment, in water, for example, and they are able to enter another human's body through the mouth by different ways, such as water, mainly drinking water, food infected by these microbes, and maybe through dirty hands, and insects such as cockroaches and house fleas. They are able to carry microbes on their legs. Usually ways, you know, them fecal oral route. But when they enter through digestive tract, but then they go to the bloodstream, and in time of viremia, short period of viremia, it is possible to be infected through the blood transfusion or maybe sexual contact. It is really, but it is possible. It's not impossible. It is possible if this blood is taken from this patient's body when in time of viremia, and sexual contact just in time of viremia. Primary defeat of children and adult persons of young age is characteristic. Children are sensitive to these viruses, and the infectious processes caused by these viruses always sharp, not long lasting, usually proceeded favorably. These patients recover and without formation of carriage of viruses. So no chronic stages, no carriers, no chronic forms. And these viruses have no oncogenic properties. Hepatitis B virus. Hepatitis B virus, HBV. Many people are infected by this hepatitis B virus. Geographic distribution of hepatitis B on this picture. And this bright color means high risk area, including Asian countries, and Africa, many countries of Africa, Middle East, and some countries on in South America, and North of America. So, and moderate risk area, including Russia, including India, and this North of Africa, moderate risk area. Anyway, many people are infected by hepatitis B virus all over the world. Taxonomy. Hepatitis B virus belong to Hepatnaviridae family, genus Orthohepatnavirus. It is Latin, don't mix with English. So morphology. Nucleic acid type double-stranded circular DNA. Pay attention, among hepatitis viruses, the only hepatitis B virus has DNA. Other viruses have RNA. So this one, hepatitis B virus, has DNA. Symmetry type spiral. The virus is enveloped, and inside the virus is own enzyme that is necessary for virus replication, and it is named DNA polymerase. And no hemolytic and no hemaggglutinating activity, no hemagglutinins. It's important that if virus has own enzyme, most of viruses have no any enzymes, but some viruses have enzymes, those are necessarily for reproduction. And it is important for pharmaceutical faculty students. If virus has own enzyme necessarily for reproduction, of course, chemicals are produced for destruction of this enzyme. Of course, it is produced. I told you that we have no so many medicines for treatment of viral infections, because the virus particle, if it is out of the host cell, there are no any metabolism. Viruses have no any metabolism, so no any target of action. And viruses are resistant to antibiotics by nature because of they have no cellular organization and they have no any metabolism. They are just able to reproduce inside the human being cells by using of ribosomes and enzymes of human being cells. If we will interrupt virus replication inside the human being cell, we will interrupt the multiplication of human being cells. So it is dangerous. So we have just a few medicines for treatment of viral infections, chemicals, and of course, immunobiological preparations. So in this case, it is possible to destroy this DNA polymerase. Lamivudine is used for treatment. Then, morphology, structure of viruses. Really, this is electron microscopy photo of the blood of the patient with hepatitis B, and hepatitis B has different shape. Many particles, sometimes looking like stick shape, other shapes, and among them there are particles detected by Dane. The scientist's name is Dane. So, and exactly these particles are infectious particles, and they are named Dane particles. Dane particles, so DNA polymerase, and there are another particles, HBS, another particles like stick shape, for example, and it has spikes on the surface. The hepatitis B virus has 4 antigens. 4 antigens. They are very famous, and you should memorize them: HBS antigen, HBC antigen, HBE antigen, HBX, and DNA is antigen 2. It is weak antigen anyway. And there is scheme of antigenic structure. Antigenic structure of hepatitis B virus. This is DNA. This is DNA polymerase. And it is surrounded by capsid, protein capsid, and it is envelope. And in envelope, there is surface antigen, HBS, it is surface antigen. And in reproduction, during reproduction of this virus, excess of HBS antigen is produced, and it is circulated in blood in free state, not connected with virus particle. And it was detected that this virus is protective antigen. Protective antigen causes development of antibodies, production of antibodies by immune system of human beings. And these antibodies are able to protect the human's body from the whole virus. And of course, the vaccine is prepared of this antigen, pay attention. And the hepatitis HBC antigen, it is core antigen. It's located in capsid and it's not circulated in blood in pre-stage. Hepatitis E antigen, it is possible to detect in blood, and not in this picture, but there is the first one, HBX antigen, and detection of it is written here, and detection of HBX antigen in the blood indicates about malignant transformation of liver. So the sign of hepatocarcinoma, liver cancer development. Cultivation. It is difficult to cultivate hepatitis viruses, not just this one. It is difficult to cultivate any hepatitis viruses in cell culture because of the site, they don't cause cytopathic effect and no indication method to make conclusion that they are reproducing the cell. So virological method is not used cell culture and they don't reproduce in chick embryo. And it was detected that primates are sensitive, like chimpanzee. Like chimpanzee, some monkeys are sensitive to hepatitis viruses, but of course not possible to use them, chimpanzee, in practice for isolation of virus from every patient. Apes, they are used for scientific purposes, of course, for testing of vaccine, for example. Resistance. Hepatitis B virus is very resistant, is very stable, heat stable, and it's still not possible to, it's still infective in spite of boiling during 20 minutes. So it is resistant to 100 degrees by Celsius during 20 minutes, resistant to alcohol, phenols, sensitive to chloramine, formalin, and ether. And it is also to destroy this virus even in autoclave during 20 minutes, and atmosphere pressure should be more than 1.5. Epidemiology. The source of infection is a person, just human beings infection, and the virus is excreted in various biological fluids such as blood, bile, breast milk, sperm, and the susceptibility of people is high. The infectious dose of the virus is very, very tiny. Contained in, it's enough, the amount of viruses to infect another person is enough in 0.0001 milliliter of blood. This amount of blood is less, more or less than 1 drop of blood. It's not 1 drop, it is less. So just traces, even the traces of blood on some equipment is enough to infect the person. Transmission routes. The disease is transmitted by the parenteral route, by passing. Parenteral means avoid the digestive tract, avoid the gastrointestinal tract, another place of entering, not through digestive tract. Sexual contact between people, if the mucosa is destroyed, through the placenta from mother to fetus. In the acute period, up to 90% of cases, fetus will be infected for sure. Then contact with, and then if the fetus was not infected, anyway, newborn will be infected, passed through the birth channel during the delivery. And of course, it is by mistake of doctors, transfusion of blood and blood substitutes. Of course, there are many procedures to avoid contamination of people due to these procedures. Just by mistake, some really cases are developed. And organ transplants, again, very really, again, just by mistake. Donors are checked now, of course, because of, they were checked before because of hepatitis B, hepatitis C, and now donors of blood and donors of organs are checked because of HIV. Human immune deficiency virus is transmitted by the same ways like hepatitis B virus. So donors are checked very carefully. That's just due to mistake, it is possible. When using medical instruments, if the sterilization was not good, again in our country, we do microbiological control of the quality of sterilization, after sterilization. And also when applying tattoos in ordinary life, manicure and pedicure procedures, and probably, and shaving sometimes, because people, those do these procedures, they have no ideas about sterilization, and even these tools are not sterilized well, it is possible to infect people. And probably this virus is transmitted by biting of bed bugs. Recently, this idea was developed, and maybe it is true, maybe it will be finally proved and confirmed, because of bed bugs exist at certain places, and they bite patients, then they bite healthy people, and infectious doses very, very small. It's enough to infect people. So there is picture: body piercing, tattooing, and shaving, and then vertical transmission and blood transfusion, really. Acidogenesis. Hepatitis B virus with blood enters the liver and reproducing hepatocytes. that hepatitis B virus is able to integrate DNA on DNA in DNA of hepatocytes and still survive like provirus in this DNA without reproduction. So chronic infection is developed and carrier state is developed, but without reproduction of hepatitis B virus. But at the same time, these hepatocytes infected by virus, they have antigens of hepatitis B virus on the surfaces. And these foreign antigens are recognized by immunocompetent cells, T lymphocytes, T helpers, because of the main function of immune system of human body is to keep genetic homeostasis. And the first task is they check constantly cells, own cells of human body, and they're looking for changes in antigens. And of course, it is easy to notice foreign antigens on the surface, and T helpers will send message to another cells, immune cells, T killers, and there are NK cells and natural killers, and they will inject cytotoxin inside the hepatocyte if it is infected. And hepatocytes are destroyed due to this normal immune response, and it is normal immune response, faster than due to reproduction of hepatitis B virus. But time by time, hepatitis virus reproduce, of course. And then the virus is released from hepatocyte and repeated viremia occurs. And generalization of infection ending with the transition to a chronic form or maybe death of hepatocarcinomas, cirrhosis of liver and hepatocarcinoma. What complications of the chronic form are caused by chronic inflammation and liver and necrosis. The main complication are cirrhosis and primary liver cancer carcinoma, hepatocarcinoma. Microbiological diagnosis. Serological method. So memorize in diagnosis of viral hepatitis. Microscopical method is not used. Virological method is not used. Biological method is not used. And skin allergic test is not used. Just two methods are used. One is serological, another one molecular genetic. So the aim of serological method is detection of antigens of microbes and antibodies for microbes. So of course in the serum and blood plasma, viral antigens of viruses and antiviral antibodies are detected by ELISA test. What are they? Surface antigen is circulated in the blood in free state. It should be detected. Antibodies to this antigen are detected. And then antibodies to core antigen are detected. And then anti-HBe antigen that indicates about replication of the virus. And antibodies to this antigen is detected. And if antibodies to HBx antigen are detected, they indicate about malignant transformation. And molecular genetic method using PCR, polymerase chain reaction, or maybe hybridization method determine the DNA of the virus in blood and liver biopsies. As usual, specific part of DNA is detected. Treatment. For treatment, interferon alpha-2b, a natural defense protein against viral infection. Now it is recombinant, I told you. It is produced by yeast cells, recombinant interferon. Now it is available. Interferons are used widely. They are not specific, and they are used for treatment of many viral infections. So it is produced now by a method of genetic engineering. The gene from the leukocytes of human beings was replaced in yeast cell and in bacterial cells, and now bacteria, we have strain of bacteria with this gene, and they are able to produce interferon alpha-2b, and it is named recombinant. Okay, and interferon against. Now scientists did develop idea that it is better to stimulate own interferon production. And some medicines are produced, some chemicals like cycloferon, like stimulator of own interferon production. And the chemical is produced, DNA polymerase inhibitor, lamivudine. Chemical, I told you that, of course, it is, if virus has own enzyme, chemical should be produced for destruction of this enzyme. Non-specific prevention of infection during blood transfusion and medical and diagnostic parenteral manipulations. In medicine, there are many items are disposal made of plastic, like plastic syringes, plastic catheters, and so on, and prevention of the sexual transmission of infection, and the prevention of accidental sexual intercourse and the use of mechanical contraceptives, safe sex, and then through strict control of the detection of viral antigens from donors. HBs antigen is named Australian antigen because it was detected firstly in Australia. So what donors are checked, what in medicine is named, donors are checked for presence of Australian antigen, HBs antigen. And specific prevention, vaccination. Vaccine, fortunately, is developed, and it is recombinant genetically engineered vaccine containing HBs antigen. I told you that this antigen is protective antigen, and it causes development of antibodies, and these antibodies in human body, and these antibodies are able to protect from the whole virus. So the vaccines are obtained by genetic engineering on the culture of Baker's yeasts. In this case, the gene that encoded information about HBs antigen production was replaced from hepatitis B virus in yeasts, fungi, Baker's yeast, and they multiply quickly, and they produce HBs antigen. And finally, the task is to separate cells of fungi from HBs antigen. We need just purified molecules, and it is done by filtration. You did study methods, filtration method. It is very widely used in microbiology. And vaccines are produced a lot of now, the recombinant DNA. They are named hepatitis B vaccine. And like mono preparation, it was used and it is included in national vaccination schedule. In Russia, for example, the first vaccination is done at the age 0. Age 0 means a newborn has no 1 day. Just the first injection is done at 12 hours of life. Exactly, first day of life, and the newborns are vaccinated. It was used like mono vaccine. Now associated vaccines are produced, and then revaccinations, a few revaccinations. People should be vaccinated, and a few times. And now this hepatitis B vaccine joined with another vaccines like against tetanus, diphtheria, tetanus, pertussis, hepatitis B, and then against Haemophilus influenza. So a few vaccines are joined now because of immunologists did prove that it is better that if the person will be vaccinated by a few vaccines at the same time, the immune response for every vaccine will be more stronger compared with using of mono vaccine. We should believe them. So hepatitis D virus. Hepatitis D virus nowadays not classified by old classification. He was named Delta virus. Now is again not classified. Classification is revised every 10 years by virologists, by international committee. So nowadays it's not classified. Morphology. Nucleic acid, positive RNA, spherical virus, envelope, and cultivation, again, difficulty, just in chimpanzee. Chimpanzees are sensitive. Antigens, RNase antigen. It has core antigen, HD, core antigen, and it has HBs antigen in outer membrane, in envelope. And really, this antigen belongs to hepatitis B antigen, but hepatitis D has got this antigen in envelope. On one serotype, no differences in strains. And epidemiology, the source of infection is the person infected with hepatitis B virus. Maybe sometimes patient is infected at the same time by hepatitis B and hepatitis D viruses, co-infection, or maybe firstly patient has hepatitis B and then he will be infected by blood of the person, those that is infected by both viruses. So why we are taking together? Because of hepatitis D virus is not able to reproduce without hepatitis B virus. It is defective. So no patients with monoinfection hepatitis D, no patients. Maybe patient is infected by hepatitis B only, but sometimes patient is infected by both viruses, B and D, but not just by D, because D virus is not able to reproduce without B. So it has HBs antigen in envelope. Transmission routes, main parenteral again with transfusional blood and blood substitutes when using medical instrument after insufficiently effective sterilization. Distribution of HDV infection, red color high distribution in South America, in South Africa, and in intermediate yellow color, and then low green color, and very low brown color. So no data. Other countries, no data about HDV contribution. Pathogenesis. Hepatitis D virus enters the liver with blood and multiplies in hepatocytes. Cytolysis by immunocompetent cells of hepatocytes occurs again, and the virus is released and repeated by VMA occurs. Microbiological diagnosis. Molecular genetic method is used. The virus RNA is detected using PCR, and then serological method, antibodies to virus antigens are determined by in blood serum by ELISA test. Treatment, interferon alpha 2b is a natural defense protein, and the specific prevention not developed against hepatitis D, but prevention about hepatitis B protects against hepatitis D. So if hepatitis B virus doesn't reproduce in patient body, not possible to virus hepatitis D reproduce. Non-specific measures are the same, like for hepatitis B, prevention of infection during the blood transfusion, donor should be checked, and hepatitis C virus. So now I will make a break and I will send the link again in 10 minutes.
Okay, thank you.
10 minutes break.
Окей, Виктор.